Hi, this is Amy Proal with the PolyBio podcast. And my guest today is Dr. Tim Henrich. He's an associate professor of medicine at the University of California, San Francisco. And there he runs a lab that studies the HIV disease process and the development of treatment options for HIV patients. And when the COVID pandemic started, Tim immediately turned his expertise on viral infection towards the study of the SARS-CoV-2 virus and its chronic consequences. He's a co-principal investigator in virology for the long-term impact of infection with novel coronavirus study program at UCSF, the first rigorous prospective study in the US and perhaps globally focused on studying the biology of what is now known as LongCovid. And Tim is also an expert in ImmunoPET-CT imaging, a form of imaging that allows his team to map deep tissue reservoirs of viruses and T-cell activity in patient tissue, which he is now doing in patients with long COVID. With that, Tim, thanks so much for joining.
Well, thank you, Dr. Prowl, for having me on.
Of course. Well, Tim, I want to begin by asking you about the imaging I mentioned, the ImmunoPET-CT imaging. And to set that up, I would say that one of the top, as you well know, when we're studying the long COVID disease process, people who develop chronic symptoms after COVID, one of the top possibilities for why they develop symptoms is maybe at least some patients don't fully clear the SARS-CoV-2 virus, and it may persist in what we call a reservoir in their tissue. But that's key, is that the SARS-CoV-2 virus may very well be persisting in deep tissue locations of patients. So the central nervous system, the brain, other body sites. So how can we study that? And that's what your imaging can do. Can you explain to me how you can image deep tissue reservoirs of viruses? I know you've done it with HIV. So if you want to talk about that, you could bring that in, or basically how you do this.
Sure. No, this is a great question. And I also agree that one of the top thoughts of why people may be having persistent symptoms over time is because there's residual virus in these tissues, whether that's protein, full replicating virus, or just nucleic acid or genetic material of the virus that's residing in tissue. We still don't know yet, but I think it's certainly very plausible that there could be some type of viral persistence within deeper tissues that are very difficult to biopsy or to just in order to characterize in participants without invasive procedures. And I think certainly we need to be moving towards ways that we can look across the entire body, not just in the peripheral blood or in limited pieces of tissue from the gut or from other places where people have looked so far. But we really need techniques to be able to probe deeper into the system and body-wide tissues where virus might be persisting. And so we've been very interested in looking at technologies that can do this with minimal invasivity for participants to make it a painless but also very revealing process. And so what we've been doing here at UCSF, I've been working closely with my collaborator, Dr. Henry Van Broeklen, who is our director of radiopharmaceutical research here at UCSF. Is to use antibodies. These are monoclonal antibodies that are specific to viral proteins. So for example, we've already done this in the setting of HIV persistence using a broadly neutralizing antibody, which is a type of monoclonal antibody that binds to the envelope or the outside part of the HIV virus. And what we've done is we used this antibody and we have tagged on a radioactive atom. Now it's low-level radioactivity. This is not high-level dangerous radiation. This is what we would use for clinical scanning for certain things like cancer or infection in the hospital. But what we do is we take these antibodies and we essentially radiolabel them. We put on this tracer that essentially emits positrons that we can identify in PET scanners, or these positron emission tomography scans. And what we did for HIV, which is a chronic viral infection, so people with HIV can take antiretroviral therapy, and despite decades of therapy, we know that as soon as people stop, HIV rebounds very quickly within a few weeks. So we know that there are these deep tissue reservoirs that harbor this latent virus. Actually, it integrates into the host chromosomes and into the genetic material of the host, unlike SARS-CoV-2, which does not do that. So I want to just say that up front, not to worry. But HIV is a retrovirus that can do this, and it can hide out in these cells. But there's been a long discussion about, is this virus active? Is there protein production? Are there viral particles being produced, even in the setting of antiviral therapy for HIV? And so the first study that we did was using one of these radiolabeled antibodies that's specific for a part of the HIV virus. We injected these into participants, and these are people with HIV, most on suppressive antiviral therapy, so they don't have detectable viral loads, but we know that HIV persists somewhere. We also enrolled uninfected control participants to help us understand what we're seeing, signal over noise, et cetera, when we do these types of assays. And we also had participants that were viremic at the time for whatever reason. Now, obviously, we don't want people to be viremic, and we always ask that people be on therapy, but, for whatever reasons that, life brings, we were able to get folks that were off of therapy, at least transiently, to see if we could see any differences in those people as well. And what we saw is that using a very, very small amount of this antibody, this is about maybe 1/100th or even less of a full treatment dose of what we would use a monoclonal antibody, for example, for HIV prevention or for SARS-CoV-2 treatment during acute infection. And then we look with the PET scanner over different periods of time. We do a single injection of this antibody, which can last in the system for several weeks. And this tracer that we put on, this zirconium, is radioactive half-life is about 3 days. So we can actually follow this up for a week, if not longer, over serial scans with a single injection of this antibody and see where it goes. And it's a really interesting technique. We didn't know what we would find when we first started this. We thought, is this going to be sensitive enough to see, potentially very, very small amounts of viral proteins that are circulating and or residing in tissue, most importantly. And what we found was, yes, that we actually saw increase in signals. We saw these monoclonal antibodies getting stuck in places like lymphoid tissues in the gut, lymph nodes throughout the body, in other places like the bone marrow, which was a bit surprising as well, the degree that we saw, in other locations in the nasopharyngeal lymphoid tissue, or this immune-rich tissue up in the nasopharynx in the back of your nose. And so we saw this light up, not just in people that were viremic, but also, although to a lesser extent, in those people that had been on years of suppressive antiviral therapy, suggesting that there is some activity, that there is at least a component of viral protein that's being made, even in the setting of that. And so that was really exciting. And that's obviously work that's ongoing in a different part of our lab. But we're certainly very interested in now applying this to SARS-CoV-2 with the exact same question. Is there persistent virus in places that we can't access by biopsy? We can't take people's spleens out, for example. It's very difficult to get a liver biopsy. Bone marrow biopsies are not comfortable for participants, and most people would not want to have those. And even if we do biopsy those tissues, it's usually only a very, very, very small piece or small pieces that we get. I mean, just fractions of the entire tissue of interest. And so if you think about the gut, 20 feet long, but when we do biopsies, we get, tiny centimeter pieces, maybe 4 or 5 of those to today take a look. So, it's just a fraction of what we would normally see. And so the strategy is to use commercial grade monoclonal antibodies that have been used to treat the original strains of COVID. They no longer work for the Omicron variants, unfortunately, for treatment. But if people infected during those initial waves through the Delta wave, have persistent virus, then we would be able to see potential proteins using this PET imaging technique, very much like we did for HIV. So the theory is that we label, and we're actually doing this right now. So we're currently in process of labeling these antibodies with this long-acting zirconium, mildly radioactive tracer, and then with the thought that we'll inject these into participants with and without symptoms, and see if we can see increase in uptake in those individuals.
Wow. Yeah, it's an incredible way to try to figure out if people have virus in tissue. And I'll just reiterate. So what you're doing is you're taking a monoclonal antibody. This is for SARS-CoV-2, just one of the ones that came out when people were getting treated during the original or Delta waves. You basically tag it with a little radioactive particle safely. Then you inject that into the subject's body. It diffuses throughout all their tissues. And then you put them in a scanner. And then the scanner, what happens, I think, right, is that the monoclonal will bind the spike protein of the virus if it finds it, and then it emits a little photon of light. And is that correct? And then the scanner sort of can see that photon and that's how the scanner maps it? Is that how it works?
Almost, yeah, very close.
Okay.
So instead of a
Okay.
photon, it's actually a proton.
A
But
proton. Okay.
similar concept. It's a very
Okay.
small subatomic particle that flies out from this
Okay.
radioactive decay
Okay.
of the tracer, and then we detect this on what we call the PET scanner, which is a
Right.
three-dimensional scanner that
Yep.
essentially, I mean, you don't see this happen when you're in it, but it circles around the body and allows us to see exactly where those protons are being emitted in 3 dimensions. And these scanners also have either a CT scan or an MRI scan built in, so we can get high resolution imaging of the tissues like we would normally do for
Right.
a clinical imaging study. So we can see exactly where this PET data are coming from. So for example, we can see where those antibodies are superimposed on these 3-dimensional images in the PET MR or the PET CT scanner. And we can also get information about any structural damage to the lungs, for example, with the CT scanner that's combined. So we get all this information at once, but that's exactly right. So after injecting the tracer, we wait a few days for it to percolate and to bind to spike protein that may be persisting in these tissues. And if it binds to that spike protein, it'll get stuck in that location. Whereas if it doesn't bind, it kind of gets metabolized and passed through the liver and eventually excreted. And so we're able to see differences in how much uptake in these different tissues. And that tells us a relative level of how much viral protein is in that area. And we can do this in very high resolution 3 dimensions using these PET-MR and PET-CT scanners to really kind of localize where this signal is coming from. And so we're very excited to get these into people quickly.
It's amazing. And one of the scanners that you're using for this study is called the Explorer scanner, right? And this is just a super high resolution. Is this the most full body? Could you explain the Explorer? I mean, and part of your, some of your team members have even just created the Explorer, right? That's how cutting edge you guys are. Is that the case?
Well, we've been involved with we collaborate with the folks that have one of the first human Explorer scanners here in the US. Which is really great that we're really on the cusp of this technology for these types of studies. And I should apologize, these are positrons, not protons. So positrons are these subatomic particles that fly out and we can detect on these PET scanners. But yeah, so this Explorer scanner is a relatively new platform. And what it is it's essentially many PET scanners put together all in one. And what that does is you can do this massive parallel imaging to increase the sensitivity and increase the signal to noise of the scan that you're doing. It also has a CT scanner that's built in as well for all this 3-dimensional tissue localization. And you know, there's some early studies with our collaborators at the UC Davis, just up the street from us, depending on traffic, how long it takes to get there from San Francisco, but could be an hour, could be 2 hours, could be 45 minutes. But either way, we've been working with them to do the very first studies using one of these monoclonal labeled antibodies, actually for HIV and now with SARS-CoV-2. And so, you know, it's really exciting because we can get about maybe 40 times more sensitivity, which may be required if we're looking for very low amounts of persistent virus. You know, if SARS-CoV-2 is persisting in tissues or making proteins in tissues, or even potentially replicating in tissues, then it may be in very low levels. This is not going to be a very high level system. This is going to be analogous to HIV, where people on therapy will have very low levels, but that low level can still set off an inflammatory cascade that can have clinical consequences. So although it's low level, doesn't mean it's not insignificant. I think that's important to note. And so the Explorer scan will hopefully help us to have even higher sensitivity to identify those areas where this low level viral production is within the body. So it's super exciting. And our colleagues at UC Davis have been really very collaborative in kind of sharing their technology for some of these seminal studies.
That's great. And then you're doing the same thing with T cells in LongCovid too, right? So you're actually you can take it. This is what's extra cool about what you're doing. You can, as long as you can tag something with a radio particle that the PET scanner can image, right? You can choose different, I mean, as long as Henry on your team or your team members can do that, you can actually also make a what ends up being called a radioligand for T cell activity too. And you've done that, right?
That's right. We have already done that, and we've actually already imaged many people with and without long COVID symptoms, and we're analyzing that data now, that initial batch. So we're very excited to see what we'll find. But yeah, and we've been doing this with HIV as well, and that what we're using this instead of being a monoclonal antibody, which is specific for a viral protein, for example. What this is a small molecule that is selectively taken up and retained by activated T cells. These are both CD8 and CD4 T cells. And T cells are a part of the immune system that's intricately involved and extremely important in antiviral immunity. So they will traffic and become activated in areas of active viral infection, for example. I mean, other things such as inflammation as well, but they are one of our main antiviral adaptive immune responses. And so what we've done is we can use this small molecule called FRAG, that's made by Cellsite, a company here in San Francisco. And when we inject this into the vein, this actually has a fluorine, radioactive fluorine, and not a zirconium. So the half-life is very short. It's just a few hours before it disappears. And so what we do is we inject it in about 45 minutes or an hour or so after injecting this. We then do this PET-CT. And this allows us not to look at viral proteins per se now, but what it does is it allows us to see where these activated T cells, the antiviral kind of immune response is located. So for example, is there an increase in T cell activity or trafficking to various tissues? Could be gut, could be the brain. This crosses the blood-brain barrier easily, so we can see if there's any immune response happening in the central nervous system. We shouldn't have a lot of T cells in the central nervous system normally. So if we see those, then that means that there's something drawing those cells into that space. And so this is really exciting because not only can we have the capacity with the ImmunoPET imaging to look at the virus itself, but we can actually look at immune responses that may be responding to the viral infection or viral persistence as well. So we're using this two-pronged approach where we could look at virus directly, but we can also look at the immune response to a potential virus infection, or even an aberrant immune response without virus. So for example, if there's autoreactive immunity or autoimmunity that develops, that we would potentially also see that with this T cell imaging agent. And so this has been super exciting because we can now use this to look essentially across the entire body in real time to see where a potential aberrant or kind of overzealous immune response may be going, which is one of the thoughts that could also be leading to a lot of the symptoms that people are experiencing after COVID-19.
Absolutely. And do you try to the greatest capacity to have the same study participant do the ImmunoPET with the tagged monoclonal antibody for the SARS-CoV-2 spike protein and the imaging that tags the molecule that can find activated T cells? Because it seems really cool if you could overlay that and say, look, this person had viral SARS-CoV-2 reservoir here, 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 and they had T cell activity here, here, and here too. And you could basically almost overlay that understanding.
Absolutely. So the goal going forward is to image these different modalities in the same patients or the same participants, you know, the same people. Yeah. Absolutely. And so, you know, this FRAG, this activated T cell marker only stays in the system for a few hours before the radioactivity decays and it's excreted pretty quickly. And so it's gone pretty quick. So we can image folks, and then any period of time point later, as closer probably the better, we can then do the ImmunoPET imaging with the radio-labeled monoclonal antibody, and we can get a sense of is there a coordination between these T cell responses and potential viral persistence, or is there a disconnect? Right. You know, are T cells not going to where we're seeing a persistent virus? Would suggest that it's not just an overzealous response, it's a misdirected response, and we're actually not getting to the compartment that we need to get to. So we don't know which one that is, but hopefully that these types of studies will inform, you know, let us know, you know, is this just an overzealous response to ongoing infection, or is it an aberrant, dysregulated, ineffective response to viral persistence over time? So these are the types of information that we can get from these studies.
Very important information. And then I know that you're also set up to actually collect tissue and gut or lymph tissue from even potentially the same study participant that underwent the monoclonal imaging and the T-cell imaging to actually verify via the biopsy if you also might find SARS-CoV-2 spike in the tissue biopsy itself, right? There's almost just yet another form of validation.
Absolutely. So we've been working with our kind of clinical director of the LIINC cohort, Dr. Michael Peluso, in our group, and what we've been doing is that we've been collecting, and we'll continue to do so going forward, of course, tissues after we've imaged. So we don't want to do it directly after imaging because we don't want radioactive tissues to be low. But if we wait a week or two, it's okay. And so what we do is a few weeks after we complete imaging, we will do either a sigmoidoscopy or a colonoscopy to get colorectal tissue or potentially distal ileum, part of the small bowel as well. We take multiple pieces from the bowel, and then we can do everything from viral detection using PCR for RNA, or we can look at in situ tissue looking for proteins, and we can also look at host genetic responses and to see how those correlate with persistence of potential virus or viral parts of the virus within that tissue. And so that's ongoing. And then we can correlate that with what we're seeing on the PET imaging. So for example, the T-cell imaging agent or the ImmunoPET monoclonal antibody to look for viral persistence. And so we'll have a complete picture. We're also now moving forward with lymph node biopsies as well, where we take cells from the lymph nodes. And then we can also do the same types of studies that we do in the gut tissue to see if we see viral persistence, and if we do or we don't, how does that correlate with some of the PET imaging, and how does that correlate with the host immune responses within those tissues directly as well. So we've done this within our HIV cohort. It was very telling that, you know, in folks where we saw increase of the HIV-specific antibody tracer, we actually saw increase in cellular HIV protein, for example, in those participants. And so this is the type of work that we're now moving on to in the setting of SARS-CoV-2 that really give us a whole picture, and also to verify that we know what we're seeing on the PET scan is what we're looking for. You know, there's always a potential that there could be non-specific signal, or it could be binding to something else, or maybe it's getting trapped in a compartment for another reason. Maybe the antibody that we give that's supposed to bind to the virus actually makes an immune complex with something that's circulating and is deposited into a tissue. And so we really want to figure out what we're seeing with that PET scanning. So what we can do is, by looking directly in the tissue, although we're just looking at a tiny fraction, remember, of the entire body, whereas the PET scan can look at everything, we can at least correlate those two and say, okay, in the region where we did the biopsy, what did we see on those PET scans? And that's very important.
That is very important. That's huge. I'm not sure if I've ever seen a group that's as well set up to study tissue, just what's happening in tissue, infection, immune response as your group. It's usually the opposite, which is, we know most teams, which is okay, but most teams draw blood because it's the easiest, well, whatever, it's commonly ethically approved, like to get ethics approval to draw blood is straightforward in this, and we capture what we can from blood. But I think one of the things that's difficult when it comes to persistence of pathogens is that with a lot of viruses, studies have shown that over time, their genetic material that they tend to clear from the blood over time. It's not really the smartest place to be, and they go into tissue where they hide, sort of in simple terms. So if we don't look in tissue, you could easily come up with the wrong conclusion and say this person doesn't have the virus anymore, when really it's just not in their blood, but it still could be in any other location. And that's what your research helps to address, which is really important. Just to clarify, if we could clone you and your team, you could.
Well, that'd be scary.
It'd be scary, but maybe worth it. It depends. Maybe worth the risk. You could technically, depending on what you could iterate, you could tag, let's say, a therapeutic or monoclonal that could also maybe find another, like Epstein-Barr virus protein, or a cytomegalovirus protein, or a Borrelia Lyme pathogen, I don't know, antibiotic like you could actually do this and potentially image deep reservoirs of many pathogens. Would that be reasonable? Is that true?
Oh, absolutely. Yeah. Okay. No, this is just the tip of the iceberg. I think, although, you know, our immediate expertise are retrovirus infection, HIV, and now SARS-CoV-2, clearly, we have interest. For example, human herpes viruses, we know are persistent chronic infections that have a latent, but also a lytic or active form, and they can flip in and out of those depending on, environmental stress or other factors. And we and other groups have also shown that there might be an association between reactivation of these human herpes viruses and the development of long COVID over time. We're still trying to piece together what that is or how that's doing it, but there certainly is this significant association between evidence of EBV, or Epstein-Barr virus, reactivation and long COVID. Now, 95% of the adult population in the US will carry EBV, and some latent, mostly in a latent form for most people, but and so it's very possible that those folks, or at least a subset of people that kind of have this acute inflammatory kind of response to COVID-19, which is an incredibly inflammatory and immune dysregulating process, could be reactivating, and that could also be kind of adding fuel onto the fire, I guess you could say, in terms of kind of revving up an aberrant and dysfunctional immune response and inflammation that could kind of have longer term impact, even regardless of viral persistence of SARS-CoV-2. And so, but not only that, but these human herpes viruses, EBV and CMV, most particularly, but other ones that can cause Kaposi sarcoma, for example, or other things are of huge interest, because they reactivate in folks that are immunosuppressed, whether it's solid organ transplant or treatment for rheumatologic disease or stem cell transplant, and long immunosuppressive use, and they can have terrible clinical outcomes. People can get very sick from reactivation of some of these. In fact, with EBV, there's tumors and cancer that have been associated with reactivation, as well as many other potential autoimmune processes, and now there's a potential link with multiple sclerosis or MS with EBV infection. And so I think that these are clearly the next low-hanging fruit, that we want to look for. Where is it? How active is this virus, by looking for viral proteins that are produced within different tissues, within different situations. So absolutely, this is something of definite interest of us. If we had more bandwidth and funds, and we have antibodies that have been clinically made in a grade that's acceptable for human use. This is the big challenge with getting into people, right, is we need antibodies that are produced in what we call a good medical practice, kind of a way of that the FDA will allow us to give it to people. And it can be very expensive to make these. And so we've relied on either commercial antibodies or antibodies that have already been made for human use in other studies. But certainly going forward, this is something that we would be extremely interested in developing and applying the tools that we have developed already for these different viruses. And bacteria as well, as you brought up. There's a group I know here at UCSF that's interested in imaging TB and persistence as well. And so, it's not just limited to viruses, but any pathogen that can be recognized with a monoclonal antibody, you can do immunopET imaging with.
It's huge. It's huge. Even Alzheimer's disease now is increasingly tied to infection, and the amyloid beta plaque potentially even forming as part of the immune response to pathogens, again, in tissue though. So there'd just be a huge ability for you to bring in patients with all kinds of diagnoses increasingly tied to pathogen processes and do something similar. It's amazing. Well, that's great. So then with the imaging that you're doing is actually part of a larger study that you're part of that I mentioned before, the long-term infection LIINC. What is it?
We can call it LIINC. Yeah, I can't even pronounce it.
Long-term infection with novel coronavirus cohort. I think something like that, which is basically at UCSF, along with your colleagues. You mentioned Michael Peluso, Stephen Deeks, other team members. Yeah. You have actually been tracking patients who did or did not develop chronic symptoms after COVID since I think it's April 2020, right? And can you tell me more about LIINC generally?
Sure. Yeah, this was a bit of good timing for collecting samples and essentially establishing this type of cohort. And what happened was back in March, late February, early March of 2020, our current government at the time was saying, don't worry about it. It's not here. It's not going to be an issue. But I think all of us in infectious diseases were like, oh dear, this is coming and it's coming fast and it's going to come hard. And so we were kind of thinking already contingency planning before we had our first identified case in San Francisco, which was actually very early during the pandemic, right? Around the same time that Boston and New York started to explode. So in fact, San Francisco, we had we were the first city in the US to go into essentially a pandemic state of emergency, even before we had a confirmed case, just showing that, this was something on people's minds and thinking that we had to think forward about. Shortly after the first few cases arrived, obviously there was a large shutdown in all sectors of life, work life especially, and a lot of non-essential research at many major academic medical institutions was shut down simply because it wasn't safe. We didn't know how this was spread. We knew it was spread very easily. We knew it could be extremely lethal, especially the people with various risk factors, and it was very scary. And so there was essentially a freeze on any non-essential research, biomedical research at a lot of these institutions, including our own. And it was at that time, as we were all deciding what we were going to do and how we were going to deal with this, that Dr. Peluso in our infectious disease division approached Dr. Stephen Deeks, myself, and some other key investigators and said, hey, we need to take the resources that we do for HIV, and we need to turn this into studying SARS-CoV-2, even though the cases have just started to appear. This is something that we need to do. And I think that was really excellent foresight on Michael's part, that to think ahead. And I think what was also unique about LIINC was that, some of this was also serendipity, but I think also a lot of kind of forward thinking from Michael and other members on the team was that rather than just studying acute infection, which was hard to do, there was a lack of PPE, for example, at the time, so it was difficult to enroll participants. We were trying to minimize the number of people that went into folks' rooms, outside of what was barely, just the bare necessity for medical care to avoid spreading to other staff or other patients. And so it was difficult actually to get studies up, and there were other studies that were forming. So the idea for LIINC was to look at the long-term consequence. I think, very early on, it became very clear that this was not flu. This was not your regular, adenovirus or rhinovirus cold. This was creating severe disease. People were coming in, walking into the hospital, and hours later were on intubators in the ICU, and many did not leave. And so, anything like this just has to have long-term consequence. There's just no way that, it won't. Now, whether that would have lasted for years like we're seeing now, I don't think we would have predicted, unfortunately. But at least we were thinking, there has to be something that's going to go on, whether it's how long is the immunity going to last? That was one of the questions we were interested in, how long are immune responses going to persist after acute infection? Are people going to be protected? But in April, May, we started to see, along with other folks as well, that many of the participants that we enrolled in the study still had symptoms. They were still quite sick. They weren't shedding virus anymore. It was hard to tell initially because we didn't have a lot of testing available. But when it was, even after people stopped shedding virus from their nose or the saliva, a lot of these folks started to have symptoms, and it became very clear early that this was unique. Now, there are post-viral syndromes and post-infectious syndromes, of course, but to the scale that we were seeing this was just unprecedented. And so we quickly decided that LIINC should really be about studying the long-term impact, not just the immune response in terms of how long is it going to be protective, but really what is going to be the ultimate impact on human health. And I think this was driven by the whole team, Dr. Peluso and Dr. Deeks and others. But I think we made that decision to say, look, this is something that is going to be of utmost interest going forward. And so we really just threw everything into that to try to build a cohort of hundreds and hundreds of people that we would follow for years over time. We scrambled for funding. We applied multiple NIH grants, many other types of grants, and we finally got one NIH supplement on one of my existing HIV grants actually to support LIINC during its infancy. But we had to cobble together essentially and we devoted a lot of our own resources, our own kind of discretional funds that we have inside the university, which we are kind of our rainy day funds and things like that. We were basically pouring into getting this type of project off the ground. But we also had an ideological idea that I shouldn't say that twice, and that was that the information that we collect and the samples that we collect should be shared with everybody. This was not about us advancing our careers. This was about a major pandemic that was going to impact millions of people worldwide. And we needed to have scientific information, samples, and data available to the greater community fast. This was the most important part. And this was the entire idea of LIINC, was not to just develop our own cohort, to develop a resource that others could use. If we didn't have the exact expertise on the ground, we would find people that did have the expertise. And if we did have the expertise, we'd find people that have even better expertise. And to really kind of use this to kind of drive collaborative science and collaborative research. And I'll tell you, this is not normal in a lot of academic medicine, where there's a lot of protectionism over projects and things like that. And so it was a big frame shift in terms of the thinking and the mindset that we all had. But I think it was just the right thing to do. You can't have a major global pandemic that's going to kill millions of people and impact millions more, and not have a collaborative research base. And so that was really what we founded this to do. And it has enabled us to do studies like the PET imaging projects, for example, like the tissue projects. We're now leveraging it to do our own clinical trials for a small pathogenesis based studies, looking at different interventions for PASC, which is kind of coming online now in the near future. And it actually allowed our site to kind of become part of the Recover consortium as well through the NIH, to kind of study long COVID in a much larger systemized fashion as well. But that said, it's been a very kind of stressful but dynamic period over the last 3 years or so for many of us. We're also clinicians, many of us well, who have treated people with SARS-CoV-2. We've all been patients at one point as well, unfortunately, and so I think that just to have these types of experiences, but to be able to say, look, we're doing something that will help understand what's going on so we can intervene and make people feel better, is the entire point of the LIINC cohort. And it's kind of finally seeing this kind of come to fruition is nice to see. That's how science should work, I think.
It is how science should work. It was the right thing to do. It seems to me also the only real way to act. It's the same thing as, you are actually part of our LongCovid research consortium, which is amazing, where our nonprofit Polybio Research Foundation, we basically felt the same way, which is we were already supporting teams and helping to conceptualize work on ME/CFS, which is a condition that also begins with a viral infection usually, and patients develop chronic symptoms. And we knew, unfortunately, a little similar to you guys, we strongly suspected that people would develop chronic symptoms after COVID, because we'd seen it with so many other viruses in these cases. And so the same thing, we just moved and said, who can we support? Who can we bring in? What can we do? How can we share samples? How can we get people on meetings to just talk, right? It's the same thing of just knowing that there was this novel virus that was capable of, that's the thing we didn't know, and trying to figure out how to best study that. I think you guys have been huge with that, where you really have shared samples with so many teams. I mean, I think you've shared up to 20,000 samples, I think, when I was talking to Michael Peluso, and a lot of the papers that have come out on long COVID are your group. I mean, it's not a surprise. And so you published on—
Or use our samples,
exactly, or teams have used your samples, which is obvious because that's what would happen. But you've done work on microbial translocation, you've done work on the spike protein and persistence, you've done work with autoantibodies, and so you really have actually gone ahead. I'll see a paper, I'm like, oh look, there's a new long COVID paper, and sure enough, the samples came from your cohort, your group. And what's really amazing is that you can follow the same people over time, because that's just huge. That's one of the things when we study ME/CFS cases, is we usually get patients at one moment in time when they come in, and maybe the infection they had was years before, 20 years before. We don't have that data to know what happened in the time from when they got the infection to when we start to sample them to see what we can study. But it's not a good thing that SARS-CoV-2 and the pandemic began, but it did present this unique opportunity that you took advantage of to study people from the time they're infected on over time, over the course of years, so that you can actually watch how people do longitudinally and check off and back in with the person 3 years later. And that's going to provide so much clarity on the chronic consequences of infection that would not be possible if you hadn't set up a study that was capable of doing that. So it's pretty huge. With those different trends, so obviously we mentioned viral persistence, the persistence of the SARS-CoV-2 virus being what your imaging is going to look at, in addition to the T cells, the biopsies, a lot going in that direction. You mentioned the reactivation of herpes viruses being potentially another driver of long COVID. What else are you guys studying? And what else have you been published on, actually? What other trends in long COVID are on your radar?
Oh, absolutely, yeah. So, I mean, this is just one of many potential etiologies of long COVID. So viral persistence, and talking about, as you're saying, PET imaging, tissue biopsies are really the way to do it. The other way to look at viral persistence is actually treat viral persistence and see if people get better. And so just empirically saying, okay, let's use either a direct antiviral like Paxlovid, for example, a protease inhibitor or a monoclonal antibody, and see if people get better in a systemized, randomized controlled trial where you can really look at discrete and concrete clinical outcomes. And so that's certainly ongoing right now as well. But there's other potential causes, whether they're all downstream from viral persistence or whether in addition to viral persistence or whether they're in folks that don't have viral persistence. But these are, so for example, autoreactive immune responses. So autoimmunity developing after infection. There can be molecular mimicry between SARS-CoV-2 proteins and self proteins, for example. And so we're just finishing up a study looking at in a huge screen of hundreds of thousands of proteins or protein sequences across the entire human genome to see if there's any autoreactive antibodies that may develop, and that's something that's coming up pretty soon. We've been looking at other autoimmune processes such as anti-nuclear antibodies, which have been implicated or are implicated in things like lupus and other autoimmune diseases that obviously predated SARS-CoV-2 infection, long COVID, looking at autointerferon antibodies, for example, another thing that we have been doing and have pursued as well. But we're also very interested in some of the inflammatory outcomes. And if you interrupt inflammation, and whether that's activation of specific immune cells like macrophages or monocytes or other ones that create these chemical signals in the body that essentially drive an aberrant, exhausted immune system and lead to inflammation. And so that's another route that we're looking at to see if we can kind of figure out how that interacts with everything from pulmonary function to exercise capacity to clinical symptoms. And if we interrupt that using different and kind of novel anti-inflammatories, not the standard steroids or things that people are thinking, can we modulate or improve symptoms in people, which is really what we want. I mean, the whole endpoint is to make people feel better and live better lives. And so that's the end goal. The other thought too is, could there be, abnormal, clotting, for example, and although we're not directly with that, obviously there are people across the world who are doing really excellent research, and we're happy to provide samples from LIINC, for example, to kind of further that type of research as well. And so there's certainly, there could be autonomic nervous dysfunction, whether, what's causing that, we don't know, but that, does interrupting that or somehow enhancing repair going to help? And so, there are a lot of different, I think, avenues that could be taken. And we're taking many of those actually, again, mostly in collaboration with others, because that's how I think the best science gets done right now.
Absolutely. Yes. And I think, right, the first point that I agree with you on is that there is unlikely to be one long COVID. I don't think anyone thinks that who's studying it. There may be someone who still hasn't cleared the SARS-CoV-2 virus, and someone who hasn't cleared it and has Epstein-Barr virus reactivate, or someone that clears it but then has Epstein virus reactivate, or someone who has just an autoantibody situation, but also, so there's probably going to be mixed and matched situations in patients, and that may be part of why people don't have very often the same symptoms. There's hundreds of symptoms that people can develop. So we know there's a broad spectrum of that. I do think when it comes to those different mechanisms that are likely involved in long COVID, one of the things that will happen, it's amazing that you're moving into clinical trials now and beginning those processes. When it came to HIV, which is your base of experience, it ended up being combinations of therapeutics, right? When you treat an HIV patient, you use one medication to contain the virus and another medication to contain the immune response, or you're using them together in ways that make sense. Do you see that as the future for long COVID?
100%. That is going to be the future. We've learned the hard way many times with many different illnesses, diseases, that combination therapies are often what work the best and lead to the most robust and durable outcomes. And so, for example, even treating acute SARS-CoV-2 infection, we use single antiviral agents for short duration of period. And that's why we're seeing rebound in some folks. And, they're still working and they're still keeping people from getting sick, but we're probably not treating long enough. And if you used a combination of 2 different antivirals or 3 potentially that target different life cycle of the virus, for example, you could, there's different direct viral agents out there that target different. You could use that in combination with monoclonal antibodies if we have antibodies that don't work for the current strains, which we don't, unfortunately, that are clinically available right now. But certainly using combinations like that, but also in something long COVID, if you can disrupt inflammation at the same time that you eliminate or remove potential viral persistence, or if you stop an aberrant clotting cascade together, then you're most likely, I think many of us would agree, are going to have better and more robust outcomes, especially because this is a heterogeneous condition. This is probably not just one thing that's consistent across everyone, but this is something that is going to be multifactorial. And if you can hit a lot of these at once, then you're going to have probably a much greater chance of having a durable response. That said, you also want to minimize toxicities, right? And I think not the problem, but the caveat of doing combination studies is it's actually very difficult to do this with the current kind of structure, the FDA approval, things like that, usually we have to show safety or potential safe, maybe efficacy, but at least safety with single use first before you start using combinations. And so there's a lot more bureaucratic kind of hoops that you have to go through to do these types of studies. It's also very difficult to get companies that make these products to work together because a different company is going to make one drug and then the antibody from this company and maybe anti-inflammatory from this company, and everybody's going to want a piece of the intellectual property, and then there's the lawyers can battle this out for months and months and months. And so, so unfortunately, there are a lot of blocks to just doing these types of combination studies, although certainly these are what we would love to be doing and would love to be doing more often and more quickly.
That's what we need. We need the pharmaceutical industry to think the way you did when you were starting LIINC, to think that there has to be collaboration moving forward. I do hope that I think there are patient organizations in the space and other groups that can hopefully help maybe some of the policymakers and pharmaceutical companies really understand how important that kind of collaboration would be in changing millions of lives. Maybe it's possible to see some progress there.
I hope so. It did happen eventually with treatment for HIV, which involves usually 3 medications, sometimes more, sometimes less. But and those medications are often made by different companies, and they've managed to work it out to make combination pills, to make dosing easier for participants, increase adherence, et cetera. And so it's certainly possible. I think there just needs to be that will and the drive on that side to be able to do it. And I agree with you, having patient advocates and academics and folks really lobbying that this is what we need to be doing. So please let us do this would be great.
Definitely. And I also think it's important that we're doing research at the same time that we're doing the clinical trials, because the research will help better delineate biomarkers, help find trends in patients that can help separate them out. So, for example, we might be able to separate out people with viral persistence more from people with EBV active. Those studies and the research that does that, including your imaging, including all of the work that you're doing from different samples on LIINC, will provide more clarity for pharmaceutical companies to know what drugs to trial in what patients and what to bring in and what combinations of drugs to potentially use in certain patients, so that we reduce the risk of toxicity, or we can actually use treatments more targeted. So I do think that's what's really important here is research happening along with the clinical trials informing the research, vice versa, to keep going to get more and more specific with this. But overall, sometimes I hear people say, oh, long COVID, it's so mysterious. I'm not sure. There's no leads. Do you disagree? It seems to me like there's.
I disagree.
Yeah.
When I talk to you, we have leads. We don't have an answer, but we definitely have leads.
Yeah.
So and absolutely.
Great. Well, all right, then Tim, it's been amazing to talk to you. You have so much going on. You have such a deep knowledge of viral infection and imaging and all kinds of what you're doing. So I'm really excited to watch as you move even more research forward.
Thank you so much. It's been a pleasure to talk.